Transmission electron microscopy uses transmitted electrons through the specimen to create the image. Meanwhile, scanning electron microscope creates an image by detecting reflected electrons. As a result, transmission electron microscopy offers valuable information on the inner structure of the sample, while scanning electron microscope provides information on the sample's surface and its composition. This slide presented how the electron beam transmitted through the specimen in both transmission electron microscopy and scanning electron microscope. This slide showed the surface image and interior organelles of cells taken by electron microscopes. Finally, from the above data, the electron microscope's applications are: 1. To visualize and study cell structures of bacteria, viruses and fungi; 2. To view bacterial flagella and plasmids; 3. To view shapes and sizes of microbial cell organelles; 4. To study differentiation between plant and animal cells; 5. To study nanoparticles; and finally, to identify damaged microparticles which further enable repair mechanisms of the particles.